Okay, thank you for the kind introduction, and hopefully I can keep you all engaged with this straight after lunch talk. So as Maddie mentioned, we just launched in May, so we're very early. It's me and my co-founder, but really excited to be supported so early in the lifetime of our company by the Hertz Foundation. And so the problem we're trying to address at the Deliverome Project is targeted delivery. And this process enables therapeutics to act specifically at cells and tissues of interest. So cells express surface proteins on the outside of the cell, and these are really useful because only about 10 to 20% of the total proteins in the cell are expressed on the surface. So they can be used as handles to get therapeutics and associated payloads precisely inside the cell to act and hopefully deliver a beneficial therapeutic effect. And people have used these in lots of different therapeutics, from gene therapies to cancer therapies, antibody-oligo conjugates, and more. So that diagram looks really nice if we could get that to work as desired, on demand, in all these different therapeutics. But practically, targeted delivery is still not fully solved beyond the liver. So we found a great protein called ASGPR that helps us get therapeutics into liver cells called hepatocytes. But beyond that, these surface proteins are really hard to measure because they're in the membrane, so they're very fragile and hard to deal with, and all quite unique in their properties. And so that means that using this strategy to target many other cells and tissues has been a little bit slow and piecemeal. And so that really means that it's difficult to scale this up because biotechs and pharma have been pursuing this one by one and not in a coordinated manner. So we thought, what about founding the Deliverome Project as a focused research organization to really build this fundamental data layer that folks could use to develop therapeutics, as well as academics working on basic biology to study these surface proteins. So we founded the Deliverome Project as a focused research organization, and the concept of these focused research organizations called FROs was really proposed and formalized by Hertz Fellows as well, Sam Rodriguez and Adam Marblestone, about 6 years ago or so. And so FROs are very unique organizations because they produce public goods similar to an academic research lab, but also require large-scale effort and coordination that's difficult to achieve in an academic environment. And finally, it's a tightly coordinated, focused research team. And so we function similar to for-profit startups in that it's a small team working together on a very specific problem. And typically, these are also designed to not continue forever, but go on for a period of about 5 years to really focus on solving that problem as much as we can during that time. And so we were really inspired to start this project now because we saw myself at Flagship Pioneering in venture creation and my co-founder at Arena Bioworks that there have been a lot of improvements in biotech on developing interesting new modalities and delivery vehicles. I'm showing some examples on the slide, but many, many more are coming down. And the design of these therapeutics has really improved with technologies like AI. But that really increased the pressure on this bottleneck of targeted delivery. So a lot of these companies were working on very cool new technologies, but targeting the same small set of surface proteins that were kind of known to be relatively de-risked and useful for delivery and getting across biological barriers and into tissues. So out of the thousands of surface proteins on cells, not many of them are characterized, and only a handful, we estimate less than 0.4%, have been used for targeted delivery. So we thought, why not start this project to potentially uncover hundreds of new delivery handles and really profile the subset of surface proteins called the surfaceome that could serve as delivery handles, which we are calling the Deliverome, so hence the name. So to do this, we're using a combination of proteomics and functional genomics technologies. We're using proteomics to measure abundance of these surface proteins on different cells and human tissues, and openly releasing that data. And then we're also measuring a per cell copy number of these surface proteins. So that's really important because when designing a therapeutic, some modalities require higher copy number, such as ADCs, CAR-Ts, and some work quite well with exquisitely low copy number, but just require high specificity. So importantly, in addition to measuring the amount of the surface protein on these cells, we're also interested in measuring internalization. And this is a property that's very difficult historically to measure because it's so dynamic. So, just like a human might be walking back and forth every day between work and home, maybe not every day with the work from home era, but in different locations. Today we're at this conference. Similarly, surface proteins, even though they're called surface, they can cycle continuously between the surface and internal compartments within the cell. And there are a few different distinct compartments within this as well. And so without knowing the rates of recycling and the types of compartments within the cell that these surface proteins travel to, it's difficult to know whether they would be a good target for therapeutics. And again, some therapeutics work quite well on the right without internalization. So in some cases, we want to target therapies to surface proteins that can serve as stable markers and stay on the surface, and we don't need to get anything in. But in many cases, such as the cases on the left, gene therapies, antibody oligoconjugates, we really want to deliver that therapeutic to the inside of cells. And to do that, we need to make sure that that surface protein that we're targeting can travel effectively from the surface to inside the cell and pull our cargo in. So to do that, we're using proteomics technologies, which I don't have time to go over in detail today, but we're also using functional genomics. And I'll kind of walk you through in a little bit more detail what the science for that platform looks like. So to study this using functional genomics, we're synthesizing libraries of surface protein and then tagging them with a couple of different fluorescent tags that we can use to then detect where they are at different times. So we can first label them with fluorophores and then allow internalization to proceed at 37 Celsius and take several time points so that we can see the rate at which these surface proteins are internalizing from the surface to inside the cell. And then since we have this dual tagging approach, that allows us to detect total protein with one tag, the one in yellow there, SpyCatcher spy tag system, and then to detect the fraction that's internalized specifically with this other alpha tag mechanism. So we can remove the surface fluorophore from the surface of the cell and only detect the internalized fraction by using a reducing agent or protease. And our collaborator at the University of Helsinki has published a preprint on this from last year using a similar strategy, but not scaled up to all of surface proteins. And then once we have this system in place, we can do very high throughput screens using fluorescently activated cell sorting, where we sort cells for the ratio of this total protein to internalized protein fraction. And then once we've sorted those cells, we can sequence the barcodes and determine which surface proteins internalize as potential handles for targeted delivery. And so I do want to highlight, science is great when we show diagrams on slides, but really companies don't work without people. So right now it's me and my co-founder, Bobby Hollingsworth there, and we've pressed into service various friends and collaborators to help us move into the lab and get things started. We're buying new equipment, as well as use equipment on auction and hiring scientists this year. So if any of you know scientists, especially PhD or a couple of years post-PhD, looking for roles in functional genomics and proteomics and cell biology, please do connect us because we'd love to talk to them. And also I did want to briefly go over, before we've set up the lab space, we started doing some computational work as well to start to think about, where would generating data help the most and where can we start analyzing the public data that's out there. And so to do that, we've collaborated with Anthropic to analyze which proteins are actually on the cell surface. So this sounds like a basic question, if we're starting with measuring these proteins for targeted delivery, we should know the list of proteins that are at the cell surface. But actually, when we looked at 5 pretty well-known scientific databases of proteins, we found that there was a huge amount of disagreement within all of these as to whether it was actually a surface protein or not. So each of these 5 databases has somewhere between 1,200 and 3,000 proteins, but the proteins that were represented in all 5 was only 188. And it's really important for us to get this list right because we need to know what to synthesize for these functional genomics experiments, and when we're detecting these surface proteins by mass spec, if they're really surface or not. And so we've been using LLMs to actually determine whether something is a surface protein or not. So there's, of course, much more detail in this, and would love to kind of talk more offline after this. We have a preprint we're drafting in the next few weeks, and happy to share like early results with that from that with anyone here. But just to kind of give you a little bit of a teaser of what this looks like, we developed a benchmark of 147 surface proteins that we manually labeled as definitely surface or contextually surface, which means they may only be surface proteins in specific cell types or cell states. Often in disease states or stress, certain surface proteins can be kind of exposed when they normally wouldn't be, or as not surface proteins. And when we compared even just Sonnet, which is not Anthropic's most powerful model, without web search at all, it actually already outperformed existing biological databases on this task. And so now we're using more advanced agentic pipelines to put together detailed reports that are backed in literature and citations from full text papers on each of these surface proteins so that we can learn as much as we can about them from public data and what's published in the literature, and then add on to that the data that we're generating in-house to help future drug developers develop therapeutics using these proteins, as well as hopefully academics interested as well. So I'm finishing right on time. Want to finish by acknowledging Radial and the Astera Institute, who've provided initial funding for us to really get this project off the ground, as well as the Hertz Foundation for the Newman Galas Award, as Maddie nicely introduced at the beginning. We're especially grateful for that because it was a big leap to start this project. So I believed in this so much, I quit my job in venture creation to start this. We didn't have the funding exactly in hand and got the award about a week after that. So that was a huge initial vote of confidence to receive that award, and really helped bring along additional funding and collaborators and partners, including some of the names of people you see here on the slide. And it means a lot to me as well because David Galas interviewed me for my Hertz Fellowship interview, so I'm really grateful to receive that award in his name. Awesome.
Hey, Brandon Dukoski. A really exciting project. I was curious the extent that you're also looking at post-translational modifications and how you're setting up to look at those kinds of differences between cells as well.
Yeah, absolutely. Great question. Would love to talk more about this. We'll be doing proteomics, including completely unbiased proteomics using the latest technology. And Astral Zoom is this instrument that we'll be using. It's a $1.6 million instrument. We're receiving support from Thermo to be able to even afford to use that. So absolutely, we'll be putting out the raw data online, and would love to work with folks who have expertise in that type of analysis so that that data can be reanalyzed as much as possible.
You might have mentioned this, but I didn't notice it. But how are you going to get the temporal information about whether a membrane receptor is going to recycle and at what frequency it's going to recycle in and out?
Yeah, absolutely. Great question. So we'll be initially binding those reagents on the surface of cells and then doing a time course at 37C. So we'll take several points in that time course and do distinct flow sorting experiments to get that temporal information.
I was wondering if you could explain what your experience was as far as what it was like applying for and receiving the Newman Galas. What was that process like for you?
Sure. Yeah, so actually we were very early in the process of thinking about this company early this year. And I actually reached out to Anne just for general, Hertz network and advice. And she said, hey, why don't you consider applying? I think this is the first time that the Newman Galas Award has been given to a nonprofit. So we had to even investigate whether that was kind of within the bounds of this award. So really appreciated just, the Hertz community's flexibility on that award, as well as just the Hertz community and people like Anne being really such a point of networking and like general advice and connection, because I thought of going to her right away, even just in the early days of thinking through this project. So that definitely wouldn't have happened otherwise. And I must say that the feedback from the committee was very helpful as well in terms of how we thought about the project quite early on.
Excellent talk. Thank you so much. I'm curious, either in your LLM work or in the work you're doing in the actual cells, if you're incorporating any information about protein export that we currently know about, or is it truly just what's on the surface?
Yeah. Oh, great question. So certainly a lot of the things that the databases miss are things that are, non-canonical. So it's typically proteins might have like a transmembrane domain to be embedded in membranes, and that's like pretty easy for these databases to catalog. But one of the examples that none of the 5 databases found that LLMs found is this KLK2 protein. It's in phase 3 clinical trials by J&J. It's a prostate-associated surface antigen. So it's typically secreted, but can also be expressed on the surface. And biologists didn't know that for a while and found that somewhat accidentally. So a lot of secreted proteins can be stably surface attached and are also quite interesting for specific targeting because since it's a rare event, it might be more specific to the disease state you're interested in targeting than a surface protein that's stably on cells. Yeah.
Hi, super cool stuff. Very excited to see the data. One of the slides had transcytosis on it, where I guess you use this protein to get another protein across.
Yeah.
Is there a way you can measure that from the assays you have? I can figure out.
Yeah, that's a great question. So transcytosis, I think most people especially consider that in terms of barrier crossing, like the blood-brain barrier is a big goal of many companies to get therapeutics into the brain, which is quite difficult. Transferrin receptor has been super useful in this, and recently an approved molecule was successful there. We felt like that is quite a specific property that we didn't want to go into as a side project and deserves its own separate project. But looking at internalization is usually a first step towards getting a list of proteins that could be useful for transcytosis and barrier crossing. So we think this data will be really useful to folks in that field, but didn't think it made sense to try to set up those model systems ourselves, which is quite an endeavor on its own.
Hi, this is so cool. I'm also I'd be really excited about that data. Thanks for doing this. I have 2 questions. One is oftentimes the actual internalization of a membrane protein depends on the specific conformation change from the binding agent. Is that something that you're looking to sort of investigate? And then in addition to that, I feel like from my experience working on very different things, often in vitro models really don't recapitulate a lot of features that you find in cells in their native environment. Wondering what are the sources of materials and tissues, and I think that depends a lot on how you're generating your library.
Yeah. Okay, both great questions. Let me see if I can get to an appendix slide on this. Yeah, so the conformation is a very important point. So a lot of times these surface proteins may not internalize just at baseline, but they need a ligand that's produced endogenously or a specific conformation of a therapeutic to actually internalize, as you mentioned. So one nice thing about the way that we're producing the library is because all these proteins are tagged, we can actually play around with the tags and specifically try to induce this cross-linking. So we'll be using this reagent called that anti-alpha tag. And then in addition to just expressing that kind of monovalent single binding format, we can actually vary the affinity, also vary the format. So induce hexamerization, for example, to really try to get that cross-linking in an artificial system. So not exactly like you might get endogenously, but similar to what one might see with a therapeutic you're developing. And then see how that internalization changes with or without that cross-linking and clustering behavior that can drive internalization. So we hope this can start to get at that question, but of course there could be serendipitous things that we'll miss and won't be able to completely cover. And then on the primary cell front, we are also pursuing mass spec-based experiments. And so we think this will be a little bit more compatible with primary cells because we can similarly label these proteins and whether they're on the surface or internalized just using proteomics. So these techniques are more compatible with primary cells from patients than functional genomics technologies, which require a little bit more manipulation of the cells, but also often have less sensitivity because they're limited by what's endogenously expressed. So we hope to kind of combine those 2 technologies to hopefully get the best of both worlds. And one nice thing about being a nonprofit is we do have access to patient tissues that might not always be accessible to for-profit startups. That was one great thing about the launch, serendipitously. Since we said we were doing this, we've had some disease foundations and doctors, clinicians reach out and specify that they have tissues available that they'd be interested in incorporating into the effort. But it's an ongoing challenge that we'll be focusing on for the next several years of the project. So if anyone hears of folks with tissues or cells that are interesting to profile, definitely connect us.
Hi, Jerry Epstein. Thanks for this really fascinating talk. I'm interested in these 5 databases that don't seem to be correlated with each other very well. Why do you think that is? Are these really hard measurements to do? And are you ever going to trust a paper you read in the literature again?
Okay, so to be fair to the 5 databases, I think one thing that's really interesting to think about with LLMs is you can ask exactly the question you're interested in with an LLM and scale that up pretty rapidly. So I think one drawback of these databases is they may not ask exactly the question we are looking to answer here. So for example, the Gene Ontology and Human Protein Atlas, these are usually based on, the Human Protein Atlas in particular is based on imaging. So you might see that a surface protein is like membrane associated, but you wouldn't know if it's on the outside of the cell or on the inside, which is extremely important for therapeutic targeting because something that is just on the inside, like KRAS, for example, is a classic undruggable protein, but that shows up in several of these databases. So it's surface associated, membrane associated, but you can't necessarily tell with some of these technologies if it's on the outside or inside, which is really critical. So I think in some cases it's the measurements, as well as just, each of these databases will have a distinct biases based on the methodology used for curation or for measurement. So I think it's not unexpected to have this, but sometimes things like LLMs can help us be a little bit more unbiased and ask exactly the question we're asking, as broadly as we'd like to ask it without restricting ourselves to features that we can measure in proteins based on sequence, like, the presence of a transmembrane domain, for example.
Jeremiah Zartman, Notre Dame. Fascinating talk. I was wondering what sort of spatial and temporal resolution you're going to get with the data set, and if you can get some sort of indication of how much these proteins are also involved with membrane contact site organization or other information like that.
Yeah, that would be great. The temporal resolution depends on a little bit on how well the assays work. So, we'll do our best. And we are trying to incorporate automation quite early so that we can scale these up and get really high resolution. There are a few new technologies we're also evaluating for more spatial resolution, where you can actually image tissue slices and select surface proteins of interest, based on those images. So those technologies, again, are a little bit limited in sensitivity because they're newer, but then allow you to ask more specific questions. So we'll evaluate incorporating those, especially in later stages of the project, to validate that the proteins we're seeing are also present in more complex tissues.
Tom Khalil, Renaissance Philanthropy. I have 2 questions for you. So I think a lot of early career researchers believe that they have 2 options, do something in a university or join a startup. What advice do you have for people who might be considering a more non-traditional route, like the one that you pursued? And then my second question is it your intuition that there are a lot of biological questions where this sort of approach would make sense in terms of things like the creation of public goods, like large, open, high-quality data sets?
Yeah, great question. I think to the first question, I would say I wouldn't want folks to think of it as, oh, now there's a third option, but there is a huge continuum between these ends of the spectrum. So I would say start with what is the goal, in an ideal world. If you could see any kind of result from your research, any data set that you think is missing, what would that look like? And then think of creative ways to achieve that. And so I do think we've seen, startups do research a lot more openly than we've seen in the past. So you can, release some of your data and work openly, as you build even a for-profit startup, as well as academics, publishing results earlier, even before they've gone through the traditional journal and peer review process. So I would encourage you to, do the science that you want to do the way you want to do it, and oftentimes that will lead to finding funders and people who will come around you and make that happen. So I would say that's a little bit the story of what happened here. I was thinking of concepts that might make sense within a venture creation and a startup world at Flagship, and thought this was a huge need, but didn't quite fit the venture mindset, and would only result in a small positive outcome if backed by venture funding, because we'd have to focus in very early and wouldn't be able to release the data publicly. And so I was really excited to think about what's the biggest vision for this project. It's not going after just one disease or indication, but really opening up the field, and then maybe later we'll become users of the data in a for-profit setting only if that makes sense at the time. And then the second question, yeah, I think data sets are sorely needed in biology. A lot of folks are becoming more and more aware of this, and it's difficult to sometimes coordinate the creation of these new data sets in academic consortia. So I think many more things that are independent, nonprofit focused research organizations could create these data sets, as well as maybe partnerships. So we're excited to partner with academics as well as biotechs and biopharma who are working on this problem and don't see ourselves as kind of the lone institution or startup working on this at all. Thank you.